Okay, hi. This is now our updated video for Chapter 8. Let's get past our little animated slide here. This topic here is going to focus on the development of the nervous system. And what I want to do before we get into some of the details about what actually is going on with the cells and the organization of the brain and sort of the role of experience. We want to also talk about the role of genes here as well. So I think we need to do a little bit of maybe a primer on genetics, and we also need to do a primer on epigenetics. That's a kind of a critical theme here. So first things first, genetics. One of the things we need to think about is that what are the functions of genes within a cell, right? So if you remember some of the basics, we have 46 chromosomes in every single cell of the body, sperm cells and egg cells notwithstanding, but all the rest of the cells of the body that do particular jobs as members of different types of bodily systems, right? So we have brain cells or neurons. We have muscle cells and skin cells. We have cardiac cells and cells that make up all of the different organs, right? So pancreatic cells of different types and liver cells of different types and so forth. And so we have all of these different kinds of cells. They have a different shape and structure and function. But within every single one of these cells, they have the same 46 chromosomes. And what those chromosomes do is they contain, of course, the gene sequences, and the gene sequences, the DNA, contain instructions here that tells these cells what to do, right? These instructions tell the cells how to do things, as I have listed here, like how to make a certain kind of protein, because proteins make up many of the building blocks of these organs and cells and the structures inside of cells. That's the structural protein bit here as well. But then there are also the functional proteins, and the functional proteins do a lot to really kind of do a lot of the basic work inside of a cell. Something that we've talked about a little bit previously in previous videos and going to come up again later in the course is the idea that I think we've covered this in a previous video of Hebb's law, right? Hebb's law is the idea that neurons that fire together wire together. At least that's the shorthand version that we will use for now. And what's going on is the idea is that when two neurons are repeatedly communicating with each other via the synapse, then what's going to happen is you're going to see a metabolic change in these neurons so that the ease with which we can activate that synapse gets stronger over time. And so what is this metabolic change? Well, this could be something like growing more dendrites or growing more dendritic spines on the pre-existing dendrites, or even adding more receptors to the dendritic spines. And these are all growth processes, right? And so the idea is that we need some sort of functional process to allow more receptors to be built into the cell membrane, and that's where these kinds of functional proteins would be involved here. At the very end of the course, when we look at some of the cognitive neuroscience stuff and learning and memory, we're going to talk in a lot more detail about that sort of stuff, what those proteins are and how they actually do what they do. But for now, we just want to have it kind of in the back of our, mental checklist of things we know is that we need proteins to do these things, right? So the DNA determines how these proteins get built for starters, right? Gives them their shape and their form and their particular folding and so forth. But it also even specifies, which cells should make which kinds of proteins, right? Different kinds of proteins do different things in different parts of the body. And this is where things get tricky because, as I said, every single cell has 46 chromosomes, the exact same 46 chromosomes. So how is it that a liver cell will make certain proteins that are important for doing liver cell kinds of jobs, whereas neurons will generate different proteins that do neuron kinds of jobs? How does the DNA know, or how does the cell know which instructions are important and therefore which proteins need to be manufactured? And this is where we want to think of the concept of gene regulation. And this is part of what is known as epigenetics, right? Epigenetics is a way in which we're going to determine how to regulate the function of the genes themselves, right? And so I like this metaphor, as far as it goes, it's okay. Of thinking of it as a librarian, right? So if we think of the DNA as a whole library full of information and full of books, right? That tells the cells how to do all of these different things, how to build all of these different proteins. What we're going to have is we need a librarian that basically has a way of restricting certain sections, right? So what we're going to do is we're going to restrict a section to say, um, uh, RNA, you're not allowed to read these books, right? And by these books, I mean certain sequences of the DNA that might be useful for building proteins in the liver, but a neuron in the brain doesn't need to read those books, right? So that's the way this works, right? Is we need a way to essentially close off a whole section of the library to say this is the restricted area, don't read these books. But meanwhile, feel free to read these other ones because this will tell you how to make, neuron proteins, right? So how does this work? One example here is called methylation. And so methylation are basically, as I've described them here, chemical tags that essentially wrap themselves around the strands of DNA. And when they do this, they will cover up essentially the gene sequence, right? And so then when something like RNA tries to attach itself to the DNA, this is how that works if you remember from a biology class, right? The RNA kind of aligns itself to a certain sequence of DNA and then it copies or it transcribes the gene sequence and then transfers that or takes that information. We remember messenger RNA, right? It carries that somewhere else into the cell. But wherever there's a methyl tag, that means that that particular sequence is covered up, and therefore that's in the restricted area, and you can't read it. You're also seeing in this picture here, besides the methyl tags shown in red, but there are also these spool-like things here labeled, it says histone modification, but those spools themselves are called histones. And I think I have a little more detail right here on the next slide. So these wheel-shaped things are called histones, right? So DNA strands wrap themselves around the histones. And histone modification is the idea is that we can wind and unwind these strands as needed. And so what's going on here is that there are portions of the gene sequences that are kind of loosely wound or even free from being wrapped around the histones. And that section of DNA is accessible. But meanwhile, there's the more tightly wound sections, right? And the tightly wound sections are basically the DNA that are tucked in between the different histones there. As you can see, it's highlighted there in red again, that, it's kind of tucked in there so that if some RNA tried to transcribe any of this, it would not be able to get into those little crevices between the histones, and therefore that section of DNA would again not be readable. Right? It's also showing a methyl group here as well, as showing that some combination of the way in which the DNA is wrapped around the histone combined with various methyl tags all throughout the gene sequence will determine exactly which sections of DNA can be accessed and which ones cannot be accessed. So again, this is how we're going to achieve gene regulation. So what this means is that however this is going to work for a neuron, we need to make sure that all of the gene sequences that are appropriate for neural functioning, right, building axons and dendrites and manufacturing neurotransmitters and all of that business, that there are certain gene sequences that are needed to do that. And we need to make sure that those gene sequences are kept in the accessible regions and that everything else that maybe is more relevant to cells of the liver or pancreas or kidneys or whatever it might be, those gene sequences are not at all relevant. So those are the ones that need to be put into the restricted section here. And so histone modification and methyl tags would be a way to do that. One more way to do that is to talk about the structure of RNA itself. So what we're looking at on this slide is we have on the left a series of gene sequences, right? So we got the DNA sequences there. You got your ACTG business there. On the right-hand side, you have a strand of RNA. And so, as I said before, right, RNA is going to line itself up to the DNA, and there's a coding process that occurs, right? So the structure of the DNA then influences how we encode information into mRNA, right? But mRNA can also do something else, and we're going to call it regulatory RNA at this point. And so what we have here is, notice that there is on this image here a start codon and a stop codon. You want to just treat this as a stop sign, a start sign, and a stop sign, right? A green light and a red light. So basically, what's happening here is that the presence of these codons essentially restrict the RNA to only read the gene sequence between the start and the stop signs, right? And so anything else is essentially not going to be doing anything. It's just going to only read the gene sequence from that little section of the mRNA strand that is in between those 2 points, right? So that's, as I have described it on the slide, this is sort of like a bookmark, right? So what we're doing here is we have a restricted section of the library that's already completely blocked off, and we're not getting into that. But even in the open section of the library where we're allowed to read these books, we're still going to set a bookmark somewhere that says we only need to read this chapter of this book, right? So that's how the regulatory RNA is going to do its job, is that once it gets into one of these books, it just knows to read a certain chapter or two, and then it can do its job. Right. So again, in one way or another, this is what epigenetics is. Epigenetics is about regulating gene expression, right? So even though we have the same genes in every single cell of the body, those cells can specialize, right? So what happens in development when we are just a single-celled zygote, right? When the sperm cell and the egg cell combine. So what did I say? We have 46 chromosomes. But at conception, 23 chromosomes from the egg cell and 23 chromosomes from the sperm cell will combine, and then creates our combination of 46 total, or 23 pairs of chromosomes. And then at this point, we have something that we could call a stem cell, right? And it's called a stem cell because it doesn't have any particular specialization, right? It's not a tissue cell of any sort. It's not a neuron. It's not a bone cell or a muscle cell or a liver cell or any of that kind of stuff, right? It's just a generic cell. And so development occurs as cells begin to differentiate from each other, right? When stem cells start to become not just a pure stem cell, but they start to restrict the range of the kinds of cells they can become. This is a gradual process here. So once we start at the zygote stage where we're a single fertilized egg, then the egg divides into two and then into four and eight and so forth. So we go through this doubling process. And so for the first couple of weeks of development from conception onward, for a few weeks there, we have a bunch of stem cells, right? And then we move into another phase of prenatal development called the embryonic stage. And this is when different tissue systems really start to emerge for the very first time. And by different tissue systems, I mean clear, distinct organization of different layers that will form into organ systems, right? The digestive system and the cardiovascular system and the nervous system will all start to be in their very rudimentary form, and then they will further develop from there. So this process of cell differentiation goes through these stages that are listed here. We start with totipotent stem cells and then move to pluripotent stem cells. So what does this mean? Totipotent stem cells are basically just the raw, rough stem cells that have very little, if any, regulation happening whatsoever, right? There's no epigenetic regulation of the DNA at this point. But then there's going to be a little bit of organization that starts to take place. And this organization means that we're going to start differentiating some of the cells, and we're going to add some methyl tags somewhere, right? And this reduces the possible future states of those totipotent stem cells. So now we say they are pluripotent. There's still a lot of different types of things they can become, but it's reduced from what it was before when they were totipotent. And then further specialization happens after that, where we get the multipotent stem cells. And this is where we enter what's called the embryonic stage of development. So I said before, there are these three layers, and those are listed right there: the endoderm, mesoderm, and ectoderm. This refers to three layers of the embryo. And to translate that, that just basically means inner layer, middle layer, and outer layer, right? Derm means layer, endo means inner, meso means middle, ecto means outer, right? So that's all that this is. The endoderm contains multipotent stem cells Now, there's not a lot of specialization here, right? We don't see organs like the stomach and the intestines completely there yet, but the digestive system will form out of the endoderm. And so when that happens, we need to further specialize those cells. But at this point, what we would say is that the cells of the endoderm are multipotent. That means that they have a possible future. They could become part of, say, the tongue or the inner lining of the mouth. They could be cells that are part of the stomach or the esophagus. They could be cells that are part of the intestine or the intestinal lining or the outer intestinal walls, right? So there's lots of different possibilities for them, but they have reduced possibilities compared to when they were pluripotent or totipotent because they cannot become neurons at this point, right? The kind of processes that have converted them into endoderm, future digestive system cells, have eliminated them from being bones or brains or any stuff like that, right? The ectoderm is where the nervous system is forming. And so here we're going to see a different kind of multipotency where now the regulation processes are making them come closer and closer to becoming a certain kind of cell. They could be neurons, but we don't know yet whether or not they're going to be a sensory neuron or an interneuron or a motor neuron. They might even become a glial cell, which is another kind of nervous tissue that's not a neuron, right? So they are still multipotent. There are multiple future possibilities, but there's still restrictions that they can't become now part of your intestines or something like that until they further develop and then become unipotent, right? And then once they become unipotent, now we're talking about something that is much more specific, where we're going to say this is definitely going to be a sensory neuron or something like that. Now, this process, this epigenetic process, is driven largely by the instructions within the cells, right? So this manages to happen on its own so that brains can develop and digestive systems can develop and musculoskeletal systems can develop and on and on and on. But it can be altered. So we can talk about things like, say, stress, because what happens with stress is that this causes certain hormones that affect DNA and gene expression, and that can alter then how this process works. And it can alter how proteins get manufactured inside cells. If certain hormones can affect, say, methyl tags or histone wrapping, then it affects gene expressions and it can put certain constraints so that cells don't maybe function as well as they used to because of that. There are, of course, external toxins that can also damage DNA. So once again, we can see some change to gene expression because of that. Certainly, nutrition factors are also relevant here as well. There's evidence, for example, of what's called a thrifty phenotype. I might have that in a later slide, so hopefully I didn't get ahead of myself there. But a thrifty phenotype occurs because if in early childhood or even in prenatal development, a child is malnourished, then epigenetic changes can occur in their cells that make them sort of slow down metabolism very dramatically, right? So the idea of a thrifty phenotype then refers to the idea that your body is starting to become very thrifty and tries to save energy as much as it can, right? Even caregiving patterns, right? So we can talk about, well, there's research with rats, for example. So when rats, infant and juvenile rats, are kept in uh, sort of isolated environments where they don't get to experience normal caregiving behaviors from their mother, this influences brain development, and we can see the epigenetic changes that are happening there as well. There's limited evidence of this in humans because, again, of course, like unlike with rats, we can't do deprivation experiments with human babies, but there are studies where kids have been raised in less than ideal environments, and we can find evidence of some epigenetic changes in their brains as well. So we find some evidence of attachment related processes perhaps that can influence the way the brain is developing here. This also then means then that we need to think about the processes of brain development here. And here we are definitely getting ahead of ourselves a little bit with these terms on the bottom: neurogenesis, neuromigration, and synaptogenesis. Neurogenesis is this cell specialization process when all of these stem cells become unipotent neurons, right? And so that means we are dividing and making more cells, but we are then creating them, right? And that's what neurogenesis means, is creating new neurons. And this happens very, very rapidly in prenatal development. As I say in my developmental psych class, we're born with at least 90% or so of the neurons we'll ever have, which is in total about 85 billion. So 90% of that or so, that's a lot, right? And that means a lot of neurons have to be created mainly during the embryonic and early fetal stages of development. Neuromigration. So what's going on here? When the neurons are created, we're going to see this in a future slide, but they're created in a layer of the neural tube in the embryo, which is part of the spinal column, in spinal cord, I should say. And but when they're created there, that doesn't mean they're going to stay there. They have to migrate to some part of the nervous system, right? If maybe a nerve branch is growing off of the spinal cord, and so as these neurons are created, they migrate down that nerve branch to extend the nerve branch as the limb itself. Maybe your arm or leg is growing, so as the arm or leg grows and the nerve grows with it, then we have to send neurons out there to populate the skin and so forth. Be the sensory neurons, the touch neurons there in the skin. But also the brain needs to grow. And so a lot of them are going up the spine and moving to form the brain. And there's obviously going to be a lot of neurons forming there. There are specialized glial cells that essentially lay a track for this, right? A track work essentially where they create this ladder-like system or a lattice-like system, and these newly born neurons will migrate all the way up down these different pathways, following chemical trails that are emitted by the glial cells. So the ability of neurons to sniff out this chemical trail that they have to follow, so they know exactly where they're supposed to go. This is also um, implicated with the say regulatory RNA that I'm describing here, right? And then once those neurons get into place, they have to start branching out. So when neurons are created, they don't look like this kinds of neurons we've been looking at in pictures in previous chapters here with, big cell bodies and long branching dendrites and axons, right? They just are a cell body at this point, and they don't start building axons and dendrites until they migrate to their final destination, maybe somewhere in the brain. So then they start branching out and they start looking for neighboring neurons to connect and hook up with, and that's called synaptogenesis. Start forming new synapses now. So the ability to make new neurons, for those neurons to migrate to the right locations, and then once they get into those locations to start branching out the way they're supposed to and form synapses, any one of these stages can be disrupted by one of these epigenetic inputs, right? In particular, we're seeing that it could dysregulate the regulatory RNA process of getting those book chapters read correctly, right? And so that means that if we see some alterations so that in prenatal development, a child does not make enough neurons, or if neurons migrate to the wrong place, and so one part of the brain ends up being overconnected while another part becomes underconnected, or at least with altered synaptogenesis, one part of the brain ends up overconnected because of too many synapses or underconnected with too few, then we can associate this with any variety of neurodevelopmental disorders, ADHD, autism spectrum disorder, and so on and so forth, could all potentially be connected to these kinds of epigenetic influences that affect how the brain develops here. This is part of what we'll call experiential epigenetics, right? So the basics of epigenetics is the idea that these natural processes of stem cells becoming more and more specialized, that's a natural unfolding process. But experiential epigenetics refers to how environmental factors also can become involved in this process. This is a part of research that looks at what we're now calling the exposome. You look that up if you want. But the exposome refers to everything that we've ever been exposed to in our lives, right? When we think about doing genetic research, you'll hear people talk about studying the human genome and decoding the human genome, which of course is an important thing to need to do scientifically. But the genome doesn't necessarily determine 100% of what happens to us as we develop and the person that we become. There's also the environment, right? It's the nature and nurture effect. And so the exposome refers to everything we've ever been exposed to in our lives. And that means possible toxins and pollutants and things like that. But it also means nutritional issues, and it just means the social environment that we grow up in. It's everything, right? It's literally everything from the basic chemical exposure to to food, to social environment, to climate, anything you can think of as part of our exposome, right? Even exposure to green spaces versus living in an urban environment. These are all differential factors that are heavily studied now as part of exposome research. It's difficult to map all of this out, but the idea is that we want to understand exactly how that works, right? And the way it's going to work is it's not just that these things affect how we think about things, right? You're talking about our social environment influencing maybe our personality traits and how we think about the world. Sure, there are these cognitive changes, but there are also epigenetic changes as a result of some of this too, and that's going to affect core brain function in many ways, right? Couple of examples that I have listed here. So in prenatal development, folic acid can be pretty important. You'll see it used as an additive in prenatal vitamins that mothers take, or even as an additive to commercial baby formulas, infant formulas. But it's really important for boosting thyroid function, and therefore it boosts overall metabolism, and that helps, of course, generate more energy because growing, especially growing brains, but growing in general is very energetically demanding. So we need to have good metabolic functioning. But folic acid seems to be especially important for growing bigger and healthier brains. So ensuring that we have a sufficient amount of that in prenatal development and early infancy is pretty critical. But that's at least one example. There's lots of other nutritional factors that are important for that. I mentioned hormones before. So stress hormones, right? Cortisol is something that is known to affect the brain. If it affects a person over the course of their lives, of course, if we're exposed to chronic stress in our adult lives, it can have negative effects on the emotional centers of the brain and even the memory centers of the brain. But in prenatal development and even infancy, maternal stress means that mom's stress hormones, cortisol and epinephrine, can also potentially influence the developing embryo or fetus and cause some of these epigenetic changes. And then there are the toxins, right? So these are called teratogens when we're talking about prenatal development. A teratogen is any sort of agent that causes maybe something that we would call birth defects. We might be interested in behavioral teratogens that affect the brain. So something as basic as, say, alcohol exposure, right? Fetal alcohol syndrome is the result of exposure to alcohol prenatally. So alcohol is a teratogen. And so in this case, that would be another example here. So it is causing changes to how the neurons develop and how the brain develops. And again, that's an epigenetic process here. I've mentioned the caregiving stuff before with the rats on the previous slide, and with humans, again, some evidence of that. This relates also to socioeconomic status, right? So the idea is that socioeconomic status correlates with a lot of different brain developmental factors. It correlates very strongly to neurogenesis. There's a note right here below that here. So what is this experiment from Evans and Schamberg, a few years old by now? Children of people who are chronically stressed have lower IQ and lower neurogenesis, right? What's going on with that? Is this because the people who are chronically stressed maybe are stressed because of their low IQ and therefore they have low IQ children? No, that's not what we're talking about here, right? What we're talking about is the idea that the parents who are chronically stressed have created an environment that is stressful. That then increases the stress level of the children, and the stress hormones in the children are influencing epigenetic processes in their own brains that reduces neurogenesis and other aspects of neural function. Their brains simply just don't grow as effectively then. And then when they grow up, with they have a perhaps lower IQ, but then potentially what we're also looking at then is that as it correlates with income level. Why? Because financial issues are a major cause of stress for a lot of people. But you can see how this might become a vicious cycle of sorts, because now people who grow up with these kinds of cognitive impairments may have less earning potential and they have more stress, and then their children have more stress and even lower IQ and other areas of brain issues. So this could go on for a while here. So this is potentially a highly problematic kind of discovery here to show that this is a pretty critical factor that influences how our brains can develop. Related here is this other study from Yehuda et al. What they found is, again, some methyl tags here on a gene called FKBP5. And we don't need to get into too much about this gene other than what I have noted here, which is that one of the functions of this gene is it regulates a specific protein that builds cell receptors for the stress hormones themselves, right? So cortisol, epinephrine, these are the stress hormones. There has to be receptors on cells, both in the brain and in various other peripheral organs, like, say, the heart and the lungs and so forth, that respond to those stress hormones. And the idea here is that they're looking at descendants of Holocaust survivors from World War II. So children, grandchildren, great grandchildren. And what they have found is that people in this group had altered expression. My light just went off there. It's on a timer. They had altered expression in that particular gene that regulates how those cells respond to stress. So you could then imagine that if we have some dysregulation of this gene, that means that we have some dysregulation in how we are building these receptors for stress hormones, which means that our ability to react to the stressor is in fact dysregulated at this point. So what does that mean? Does it mean, as I have it suggested down here, that gene disruption of that particular gene is associated with post-traumatic stress disorder susceptibility and major depression susceptibility? Yes. Now, it doesn't mean that being born as a descendant of any one of these people who were survivors of the Holocaust determines that you will have some sort of mental health concern like this. But we certainly see that the presence of that genetic alteration influences susceptibility is a huge finding here, because what it's really showing is that these epigenetic changes are not necessarily things that happen to us in our own lifetimes, but these can be things that happen to us in our not to us, but can happen in our parents' lifetimes or our grandparents' lifetimes. And these kinds of things can be passed down from generation to generation. I should emphasize that there is a little controversy about these findings into whether or not, these got these two studies here from Evans and Schamberg and Yehuda, and there's not a whole lot more of these kinds of studies, but these are certainly provocative findings that are making a big deal in epigenetic research here. Okay, I think that's a good place to stop, and I'm just going to run back to the next previous slide here. Before we get into the next section, which focuses on the details of what's happening at a brain development level, the actual structures, right, and what's going on there, we can make that another video. So I will go ahead and call it a day on this one, and I will see you at the next one.